Welcome to the Great Lakes Seminar Series, co-hosted by the Cooperative Institute for Great Lakes Research and the NOAA Great Lakes Environmental Research Laboratory. My name is Mary Algal, and I am the Program Manager for SIGLR. It is my sincere pleasure to introduce today's speaker, Dr. Jordon Beckler. Dr. Beckler is an Assistant Research Professor at Florida Atlantic University with a joint appointment with the Harbor Branch Oceanographic Institute, the Institute of Sensing and Embedded Network and Systems Engineering, known as iSense, and the Chemistry Department. As the PI of the Geochemistry and Geochemical Sensing Lab, his group's research centers around unraveling sediment biogeochemical processes in near-surface sediments and connections to ecosystem health. The lab develops and commercializes innovative technologies for monitoring sediment geochemistry and exchanges with the water column. His current projects include monitoring Lake Okeechobee harmful algal blooms, the fluxes of carbon, iron, and CDOM from continental margin areas, and the exploration of submarine sink blue holes offshore in the Gulf of Mexico. Before I turn it over to our speaker, I would like to remind everyone to please leave their microphones muted for the duration of the webinar. You're welcome to enter questions into the chat box at any time during the presentation, which I will moderate at the end of the presentation. With that, I will turn it over to Dr. Jordon Beckler. Welcome, Jordon.
Thank you so much, Mary, for the intro and for organizing this whole thing. I'm obviously excited to be here. There's just so much that we can learn by looking at, by comparing and contrasting Lake Erie versus Lake Okeechobee, which are, arguably the two most notorious algae bloom affected lakes in the country. So I also wanted to thank Casey Godwin for getting this whole thing in motion. He's my primary contact up at your institute, and so thank you, Casey. So the work that I'll present, this doubles as my acknowledgement slide. There are a lot of people involved in the project, too many to name them all. It's something like 10 people full time. So we have collaborators at Florida Gulf Coast University. That's Mike Parsons' group, at Texas A&M in GCUS. They're the ones that manage our data portal. And then we have a couple of private companies as well. NavOcean Inc. You'll see they operated and designed the autonomous surface vehicle, the sailboat, and then AIS Instruments, or AIS Incorporated in New Jersey, longtime friend and collaborator who helps out with the in situ sediment work. I just want to mention, you can follow us on Instagram if you're into that. Our student maintains our account. Here is the Instagram address. And, we're constantly posting updates about mostly this project, actually. So this work, I have to acknowledge Florida Department of Environmental Protection for initially a grant. We've had now for about 2 years through the innovative harmful algae bloom mitigation technologies program, and now they're sort of transitioning us through the Office of Environmental Accountability and Transparency. So today we will talk about just some background of the lake and the focus of our project, which is really to develop an integrated observing system using innovative technologies to try to detect harmful algae bloom ecology and dynamics, or to try and understand this ecology and dynamics, and then ultimately to input all this data into a predictive model that allows us to identify the blooms at early stages when you could potentially still do something about it. But we're also, just developing baseline fundamental knowledge that allows us to understand how the bloom works. And also this baseline knowledge can help us evaluate any sort of secondary effects if someone does decide to go out and try to actively mitigate the blooms by adding chemicals or clay or aluminum, whatever it may be. So I'll discuss the model briefly and some major requirements going forward that we've identified. I'll give some highlights from the 1 year of operations that we've had to date, basically calendar year 2021. The work that we have planned going forward, basically starting tomorrow. And then one synthesis slide that will hopefully be fodder for some discussion comparing Lake O and Lake Erie, at least the Western Basin. Just a little background, Lake Okeechobee. It's the I think, the second largest lake in the US. It receives a lot of inputs, a lot of agricultural inputs from the Kissimmee River drainage basin. You also get small input, or I should say a disproportionate nutrient load relative to its actual volume through Fish Eating Creek here. Now, the water used to flow down south through the Everglades and get filtered out, but now they basically, the Army Corps has decided, or not decided, but they've built structures that divert the water through the Caloosahatchee River and estuary. And then on our side, where we are located right here in Fort Pierce, through the St. Lucie estuary. And you, of course, get effects from the nutrients or even the algae blooms themselves, the algae cells themselves in the Indian River Lagoon here. And perhaps also, these nutrients contribute to red tide in the Gulf of Mexico. That's, certainly something that's still highly contested. So this is some data that Tim Moore, a collaborator on our project, put together. This is just the lake, sort of a picture of the average locations of the Microcystis blooms just using satellites. Here's an older satellite that was in operation from 2004 to 2012. You see the blooms here in yellow for the warmest months where we usually have issues with the blooms. Now, if you look more recently over the last 5 years or so, you see more yellow, right? So we have more blooms, the blooms are getting worse, and even the geographical patterns have changed. So in general, we kind of think, although there's plenty of exceptions to this rule, that the blooms kind of start over in the west near Fish Eating Creek and make their way clockwise, and maybe they pick up a lot of nutrients from the Kissimmee River or the underlying mud sediments, which is really what I'm going to focus today. So you do see these geographical differences, both 20 years ago, but then also more recently. And you see these differences also, with respect from month to month, which is, it's kind of strange when you think about Florida being warm, and we're certainly within the tolerance range of Microcystis, of the Microcystis algae. And so there has to be something else going on, whether it's wind, currents, the underlying sediments, something is causing, for instance, this hot spot to emerge here more recently than we see in June. So we're trying to really get at, these more underlying factors. So Lake Okeechobee is much shallower than Lake Erie. It's really just a puddle on some mud. It's only about 10 feet deep on average. So just for those of y'all who aren't too familiar with this system and algae blooms and whatnot, we have a lot of nitrogen and phosphorus that comes in, let's say from a river. Some of it can be immediately utilized by Microcystis. You can also have the sedimentation of particulate nutrients. Phosphorus in particular can adsorb to iron oxides. They're like natural sponges and they're very abundant in this system. And they would be buried, except due to microbial processes, microbial respiration, basically organisms that are using iron oxides instead of oxygen to respire, they dissolve those iron oxides and this phosphate can then be re-released to the water column. There's other pathways too, but this is the one that I think is most important in this system. Dissolved inorganic nitrogen in our lake, typically it's found as ammonium, the most reduced form in the pore waters. It fluxes outwards, but it's rapidly transformed into nitrate. That's what we're seeing, and the majority of the actual water column is the more oxidized, more thermodynamically stable nitrate form. So this is showing the last, 50 years or so. This is long-term data from the South Florida Water Management District. You see phosphate, so your conventional sort of phosphorus nutrient is, you have these annual depletion and accumulation cycles. It's being depleted over the summertime when the blooms come. But then something happened in 2004, 2005. We had really bad hurricane season, and it seems like we've had a regime shift, so an ecological regime shift, where now you sort of have your phosphorus concentrations, they're higher than they used to be in the water, despite us actually at least on paper, limiting the total phosphorus inputs, the external inputs into the lake. You don't get this sort of guaranteed depletion every summertime anymore like you used to. So, it seems like there's an excess amount of phosphorus in the lake now. Compare this to nitrate where, compare this to nitrate where you still do get this annual depletion, and you don't necessarily see a long-term increase. Carl Haven suggested that what you really have going on here is, you originally had phosphorus limitation. Eventually, you sort of overcame this phosphorus limitation, and now you have this secondary nitrogen limitation, which, just anecdotally from working on the system for the last year, I really think that nitrogen is the more important control, at least over the short term when you're looking at seasonal trends. So I borrowed this from Sean Scully at the district. These are the annual averages for nearshore and pelagic sites of all of a year's worth of data. And what you see based on the last 5 years is when you have high turbidity, so let's say sediment resuspension going on, it matches the phosphate concentrations pretty nicely. Same thing with the dissolved inorganic nitrogen. So, this to me says that you have sediment or sediment nutrients that are being resuspended and redistributed throughout the lake. But what you don't see is a necessary sort of dependence of the total chlorophyll on these relative nutrient loads. So that just says that there's more to it than just the nutrients. It could be the timing of the nutrient delivery. It could be things like light, could be submerged aquatic vegetation, lots of different things. But overall, this really does show that the turbidity and the sediment resuspension is important for delivering the nutrients to the water column. Now, there has been these decadal surveys that Fisher et al. And I know Todd Osborne's group at UF is continuing through the district. So they go out and they map the total inventories of the solid phase nitrogen, the solid phase phosphorus, but then they also look at the pore water nitrogen and phosphorus. And this is the data that I found that's publicly available, at least easily publicly available. It's a little old, from 88 to 98, 10-year period. But what you see just in this period is that you get a massive increase in dissolved pore water phosphate. And this is the more bioavailable fraction. If it is resuspended, it's going to be directly available to the Microcystis algae. I forgot to mention, these are the pore water nutrients, but the solid phase nutrients were relatively constant between these two time periods. So for whatever reason, we have more solubilization of these nutrients within the pore waters. Your pore water ammonium, it shows the same thing. You have an increase, and this is over here in the really muddy area of the lake. And this is anthropogenic, human induced mud, as you'll see from the next slide. Actually, you won't see that on the next slide because I removed it, so my apologies, but this tends to be the muddier area of the lake where you are seeing, these nutrient increases. The mud area is loaded with nutrients, and it's really, sort of like ground zero, we think, in terms of the sediment nutrient source of the lake. So our operations, we did a lot, we had a lot of folks out there. We tended to focus on a couple of these towers. The district, they installed these really convenient monitoring platforms out in the lake because there's not really that much else out there. So we installed these LOBO systems to monitor water quality. I think they're similar to the systems that are operated in Erie, at least when there's no ice cover. So we were able to obtain surface and bottom water nutrient concentrations at high temporal resolution, I think for the first time ever for this lake. We, of course, utilized remote sensing to get that wide area that wide area distributions of the algae, and I already showed that data. What else did we do? We had this autonomous sailboat, which I'll talk about. In terms of the water column, we went out bi-weekly. We collected in-water optical properties to try to basically develop algorithms to better detect microcystis, but also understand the light environment and how that controls their distribution. We had a couple of innovative HAB sensors that we deployed, which I'll talk about briefly. And then we did a lot of sediment work, and this was my group. We collected a lot of sediment cores. We looked at the near surface concentration gradient of the pore water dissolved analytes, and we can determine sediment fluxes that way. We deployed a benthic lander with in situ incubation chambers, which I'll describe how they work to measure nutrient fluxes from sediments in situ. And lastly, I won't talk about this for the sake of time, but we did deploy some really innovative sensors to try to monitor sediment respiration and sort of iron cycling in situ in the sediments for an extended period of time. So the model that we're ultimately doing all this work to try to parameterize is based on the oceanographic ROMS model. We are an oceanographic institute, so we're, in all honesty, I think we're relatively new to the whole lake realm, and I don't think I'm alone in saying that. Now, the model is high resolution, 3-dimensional, and I think it runs, on the order of every few minutes, and those are the time steps of the model, and it's able to output, let's say, a forecast, a week in advance from the day that you run it. So this is just an example of the model output for chlorophyll and nitrogen. And the whole idea is, if we could pinpoint when a bloom is going to emerge and where it's going to emerge, you could go ahead and then try to conduct your mitigation, whatever it may be, while it's still a tractable problem, while the bloom is still small, while it's still, efficient to do so. In terms of where we're going next year, major things that we still need to improve in the model. It's the diurnal behavior of these organisms, the nitrogen transformations, a sediment diagenetic model to try and understand the biogeochemistry, basically take all the data that we generated last year and try to model it so that we can easily put it in this model. And then really work on parameterizing our diffusive versus resuspension sediment fluxes. And so I'll highlight some of this stuff as appropriate going forward. Now, I only have a second to talk about this, but we did, try to validate also what the satellites were seeing by going out on the water and using a spectroradiometer, just a handheld one that's, kind of measuring what the satellites would see, the remote sensing reflectance bouncing off of the water surface. And what you see is when you compare the actual number of cells, this is kind of our independent variable based on what we measure with the radiometer or what a satellite would measure. The actual data points are very under-predicted, right? So we underestimate the number of cells using the current operational NOAA algorithm. And so Tim Moore in our group, he's working on, reconfiguring this algorithm to better fit. And the whole cause of this is because it's such a turbid lake, you can see how brown the water is. The satellites, it's really like working on a whole nother planet, kind of, almost literally. So to fill in the gaps of the satellites, because really you're only getting a usable image, on average, I'd say probably 2 days or so, or every 2 days. We had an autonomous surface vehicle, company NavOcean. It's called the Sail and Solar Drone. And it basically ran a triangular loop. I think it did it about 80 times over the course of the year. And it had water quality sensors that it's measuring, pretty generic water quality parameters in the surface of the lake. It also had an ADCP on it, so it's measuring the water velocities below it over the full water column depth. And this allows us to really look at, let's say, the shear stress that would be, or we can look at the pressure, I guess, that the sediments would be under to want to be resuspended and deliver nutrients to the water column. Now, the spatial patterns were highly variable from east to west and north to south. And these gradients are pretty powerful. When we look across any one of these gradients, it allows us to use these more basic parameters, for instance, to try to identify a species-specific algorithm based on when we actually go out and we validate, and then we look under a microscope and we say, hey, you know, Microcystis has a certain phycocyanin to backscatter ratio relative to some other species. So we're still working on trying to develop an algorithm for that. But what you do see here is the east to west transect from left to right as we go on to the year. So we start out January 1st, and as you see, we end up, this is data through mid-October, and we start seeing activity of cyanobacteria as early as February. And you know, we then see the Microcystis bloom. It pops up in April. There's a lot of white space, just because it's kind of a learning curve associated with it. But then when we get to the white space up here, it's because the sailboat is running other transects, right, in between these single lines. And so, you know, there's a wealth of data here for us to mine and try to understand what's going on. So I just wanted to show you that data is available, and it's a pretty exciting data set. We also added new biological measurements relative to what's being conducted already by the South Florida Water Management District. In particular, we measured bottom water. Every time we went out, we measured Microcystis in the bottom water. So you can see here, this is a log scale of the Microcystis cell counts. We see the bloom emerges in late April, but at this site, which is our primary site up here, it's a little bit shallower, a little bit clearer water. It has a sand, you know, a mixed sand and mud bottom. We don't see stratification between the surface and bottom of the water column with respect to the actual cell counts. When we go to a deeper area of the lake, and I mean, you know, this is like 3 meters versus 4 meters, so we're not talking too much deeper, but this one does have a muddy bottom, very mud. It's, you know, that typical anthropogenic sourced mud that's been accumulating over the last century or so. We do see a much higher density of cells at the surface than we do at the bottom. And this kind of makes sense. You know, we think that Microcystis can thrive in these turbid water bodies around the world because it can regulate its buoyancy. It can outcompete these other organisms for light. But there is a caveat in that we typically sampled this site at about 9 a.m. and we didn't get to this one until about noon. And so hold that thought for a second. We deployed also, I think this was the first time it was used for, you know, trying to detect algae blooms. It's basically a fish finder that sits on the seafloor and points upward. It's called an acoustic zooplankton fish profiler. So here's a 5-day test deployment. And what you're looking at is the water column. This is the 3-meter water column. The red line is just the surface, so ignore anything above that. And what you're looking at is the turbidity, or just the particle counts of the water column. What you see here is at nighttime, you have a well-mixed water column. We don't really see any stratification, at least in the particles. During the daytime, you do see stratification. We think that this is due to the Microcystis, you know, regulating their buoyancy, migrating up to the surface for nutrient, or migrating to the surface for light during the day, but then going back down at night to get nutrients from the sediments. Now, remember that last slide, when we typically sampled at 9 a.m. and we didn't show any stratification, you know, there you go. When we went to the other site and we did see stratification, we indeed do see it at that time. Now, granted, this single deployment is only from this L1 site, so we're not sure if the same thing actually happens at this LZ40 site. So it's a little inconclusive as to whether these spatial patterns, or let's say as to whether the vertical stratification differences that we saw spatially were due to the time of day that we sampled, or based on some other environmental factor like the enhanced turbidity. Maybe it's a little bit of both. But I think, you know, it's really important just for us to realize that, you know, we will get these spatial variability. This is something that we really have to consider in terms of our sampling regimen, and not just us, but also the ongoing work from the water management district. So something, a little word of caution, I guess. We also deployed a AUTOHOLO. It's a holographic in situ microscope that was designed at Harbor Branch. And it's collecting 3-dimensional images from the water column. We weren't sure if it was going to work because of how turbid the lake was, but it actually worked really well. We detect Microcystis colonies, copepods. And this is something that we're going to be deploying on Saturday, actually, for a longer period of time to try to get at this vertical migration story. So hopefully we'll have answers to that pretty soon. Looking at the LOBO, the in situ water quality measurements, we had one at the surface and one at the bottom of that northern primary L001 site. Now I just want you to focus here on the purple box. I know it's a busy figure. What you see here are the discrete phosphate sample, the water column phosphate samples collected when either our group, the halo group in orange, or the state goes out in blue, and we measure phosphate in the water column on samples that we take back to the lab. So here's a 2-week period, and you would assume, if you didn't know any better, oh yeah, well, it's about constant, no big change. But when we look at the continuous phosphate measurements that are measuring in situ every hour, you see during that entire time period, we actually had a full depletion and then accumulation cycle. So that, this short-term variability is obviously really important if we're trying to understand the short-term variability of the algae themselves. Now you also see something happen here in late July. The bloom dies off. This is from a Seaprism. It's basically an in situ radiometer that's measuring how green the surface of the water is. So it's measuring these surface scums of Microcystis. The bloom shuts off like a light switch in late July. We did have a tropical storm that came through on early and mid July, and then there was a few more sort of resuspension events here. And I just want you to note the timing of this because it's going to come up later. One other thing I want you to see is that turbidity, it's not that clear from this diagram, but turbidity we often saw spike along with phosphate. So it was pretty clear that these inputs are due to wind or wave induced resuspension events. As far as the sediment stuff, moving down, here's a map of the lake. You can see the mud area here and then a little bit up here. You can see the sites where we sampled cores at. I'm going to show data primarily from our main site, L1, and then L4 and LZ40 were our other two sort of primary sites that we collected a lot of cores from. We also had a little side project. We were looking in the Pahokee Marina. This was a really algae affected area. Received a lot of press, so you might have even seen the picture that I'm going to show here in a second on the news. So each core we section into 10 depths. We have 5 millimeter resolution in the top few sections. So that allows us to then calculate diffusive fluxes using Fick's law. Each one of these sediment samples, both the pore waters and the solid phase samples, undergo a whole bunch of measurements, pretty much everything you could think of in terms of organic and inorganic nutrient forms, as well as the partitioning of phosphorus onto solid phase minerals using sort of a modified Tessier extraction. Here's just the very first sampling we actually did. You see in the west, sand bottom, and in the north, a mixed mud and sand bottom receiving Kissimmee River inflow, Pasheating Creek inflow. You see how clear these sites are, or how clear the overlying water is from these cores. Compare that to the two muddy sites, the mud bottom sites, and we obviously just taking a core resuspends a bunch of sediments. So we try to be as careful as we can, but there's really only so much you can do. But it just goes to show you how easily this material can be resuspended if you have any wave or wind action. In terms of the spatial variability in pore water nitrogen, so this is pore water ammonium, we didn't really see much pore water nitrate, maybe in the surface section, the most surface-most section, but that was about it. And even then it was pretty low. So the sediments were by far and large dominated by ammonium. And that makes sense because they're mud, right? And they have high respiration ongoing in them. So at the L004 site, this sort of open water mud bottom site, you don't see nearly as much ammonium accumulating in a sediment profile down to 8 centimeters as you do in this Pahokee area, this confined Pahokee area. And the reason we were initially contacted or asked by the district to modify our sampling regimen to go and take sediment cores from this area is because they were detecting ammonium in the surface water in this Pahokee area. And they said, well, you don't see ammonium anywhere else in the lake. You really only see it in Pahokee. And sure enough, you see how much larger, this is the same scale, you see how much larger your concentrations are in this Pahokee marina relative to L004. So you certainly would have an ammonium flux out in this case when we see a near surface gradient like this. So, that could definitely explain why the blooms really took off in this area. Now, I want to show you now, that was the spatial variability. When we look at the temporal variability, I'm just showing you now one site, but I'm showing you pore water ammonium and pore water phosphate. Your ammonium, I'm using the same scale as before, so you can't really see the differences that much, but there is some change over time, and I'll kind of zoom in on that in the next slide. Now, your phosphate, on the other hand, is highly variable with much greater phosphate release in the warmer summertime months in late August. So a lot of more phosphate being released to the pore waters, likely from the dissolution of iron oxides, as I'll show you some data in a second. So, this suggests that your relative, if we're just using these cores and we're just saying that, what you get from these diffusive flux measurements using Fick's law in terms of the gradient, the near surface gradient, and how you can then infer a flux across the interface from that, you would get drastically different relative N to P loading ratios. And so that's what I have plotted here in blue. You see the ammonium to phosphate ratio over the course of a year at the site of the sediment flux. So this is the actual, sediment leaving material. You see you have a very high, it's off the chart here, N to P ratio in the beginning, but this slowly declines or quickly declines as we move on into the summer months. You see that your ambient, just your bottom water, DIN to SRP ratio, so basically the same thing, but we have to include nitrate now in this ratio. You see that it kind of shows the same thing, but it lags the sediments by about a month. So maybe that lag is related. Maybe the sediment flux, it takes some time, and then the water column takes a couple weeks to respond. You can see Microcystis, it prefers elevated nitrogen to phosphorus ratio. This is when the conditions were right for the bloom to begin, and then the bloom largely dissipated when we got to this point. So, the timing, at least based on these N to P ratios, it does seem like the sediment fluxes could explain the bloom patterns. Now, I know this is an overwhelming amount of data, but I just wanted to show you how much data we have to work with, and how much of a mountain we have to climb to try and interpret all this data. I just want you to focus on this first column, which is our primary site. And what we have plotted here is our dissolved oxygen uptake in blue. Now, this uses this axis over here on the right. But what you see is our dissolved oxygen uptake over the course of a year. It reaches a maximum in August. And this makes sense. It's warmer temperatures, more respiration, a lot of accumulated organic matter over that time. So our sediments have a very high oxygen demand at this time. You would expect that your nitrogen and your phosphorus being released from the degradation of this organic material should show a similar trend. And phosphate does. If you look in red here, this is your sediment flux, your diffusive flux calculated from sediment cores. You see for the most part, you have a larger flux here in red. The black data points, by the way, are the ambient bottom water phosphate. Now, this is the most surprising thing that I think, in this whole entire project that we sort of found. Your ammonium doesn't follow this trend. Your ammonium increases early, like when the blooms start, right, in late April. And then it's progressively, it appears like it's removed, right? Because you would expect to have the most ammonium production here, but it seems like it's really being removed over this time period. And, we don't really know what's causing this, but we have some ideas, and I'll get to that. I want to kind of show off a cool technique that we have to look at phosphorus cycling. The whole idea is that the iron oxides, they absorb phosphate. You have iron-reducing bacteria that couple the oxidation of organic matter to the reduction of these iron oxides, releasing iron 2 and releasing free phosphate to the pore waters. We do see correlations. This is in contrast to previous work. Actually, when we're doing this high resolution work, we see, or at least this high temporal resolution work, we can pick up on more patterns. We do see a correlation between this reduced iron product and this phosphate that accumulates in the pore waters, but it is a site-specific ratio, so that may have something to do with the actual phosphorus loading onto these iron oxides. Now, remember that drop that I showed you, how this precipitous drop in Microcystis at this site come late July? Our water column phosphate also dropped shortly after that time period. And our pore water phosphate inventory, each one of these data points is just a separate slice from our sediment core. So our pore water phosphate also drops at this time, but then it kind of rebounds as well. And so we wanted to know, could this have had some sort of physical effect that has to do with the iron in terms of, the iron's controlling the phosphorus solubility? And so what I plot here is the first detect depth where we detect dissolved iron. We obtain this using voltammetric. It's an electrochemical profiling technique where we're able to actually measure dissolved iron. And we see, as you get the onset of summer, you start seeing iron reduction. We start seeing dissolved iron 2 only, a centimeter below the sediment water interface. So that suggests, you have phosphate being released near the sediment surface, so it could more easily, let's say, be resuspended or even break past the iron oxide surface layer if in the event of a diffusive flux. Now, right there, when that storm comes through, this drops, this iron detect depth drops, and what we think is going on is this period right here, or this area right here, this top 4 centimeter depth increments, we think that you essentially reoxidized your iron oxides, you scavenged a lot of phosphorus, and then any phosphorus that wants to flux out is not able to because it gets trapped on the iron oxide. So you would really need, a significant resuspension event to resuspend any of that phosphate. So just showing you briefly how we did this, and I'm happy to talk about this more afterwards. We use a technique called, it's called voltammetry, and we use a custom fabricated gold electrode that we form a mercury amalgam on it. And then we can slowly insert that into the sediments. We also use a pH electrode right next door. And so we can get these high resolution depth profiles. And so here's just an example of where we didn't detect iron until, down deep, 3 centimeters or so. You see, here's a good example. In this case, let's see, you see your oxygen basically go to nothing right below the sediment water interface. This is in contrast to what you see in Lake Erie. And now in this case, you have your iron oxides, or you have your dissolved iron being detected very close to the interface. So this is a situation where you would have, that would be conducive to a phosphate flux, whereas this situation, not so much. We also see a drop in the pH in this area, which also suggests that you may have iron oxidation going on. So we think, that's really controlling sort of the micro patterns, the short-term patterns of phosphorus cycling in the sediments in exchanges with the water column. So the Benthic lander, we deployed this, I think a total of 12 times, but we really had to work out a lot of the kinks the first half of the summer. So, we basically deploy this for about 7 hours. That's all it takes. You have, you can imagine you're taking like a coffee can and you're sticking it, you're inverting it, you're putting it on top of the sediments, and you have this little parcel of water that's trapped underneath the sediments. And now we monitor as material accumulates in there, and we know the sediment surface area. And so then we can back calculate a sediment flux from that. So we had a dark chamber, or like it's an opaque chamber, and we had a transparent chamber, so we could look at the disparate effects of photosynthesis and respiration. We collect syringe samples over time from each of these chambers, in the order of every hour and a half or so. So I'm just showing 2 examples because it's a mess if I show more. But what you see is, let's say it worked, but it didn't work for the reason that we wanted it to work. When we do this work in the ocean, which is where these techniques I think originated from, you basically assume that anything that happens within the chamber is directly due to the sediments themselves, because everything happens a little bit slower in terms of the biology. When we do this in the lake, we see drastic effects if you have, depending on whether or not you had Microcystis present in the bottom water when you started the incubation. So let's take phosphate, for instance. We don't really see a phosphate flux under most circumstances, except a couple times when we saw it in the last time point. And maybe the reason for that is because by this point we had, I'm not showing the data, but we had depleted our oxygen within the chamber. The sediments are actually sucking up that oxygen. So they depleted the oxygen, by about 50% at that point. And so maybe we only see phosphate fluxes when you can induce hypoxia. Now this is a problem because in the lake, we don't really get hypoxia in Lake Okeechobee. It doesn't really happen like it does in Lake Erie. So, my gut feeling is that you barely get any phosphate fluxes in the lake unless you have a resuspension event to deliver those pore waters into the water column. Now, ammonium, this was the one that really stands out. When you don't have many Microcystis cells present in the ambient water, you see ammonium accumulate in the chamber. When you do have Microcystis present, you don't see ammonium accumulate in the chamber, likely because they're rapidly taking up that ammonium. So even just a few cells, are able to suck up a significant, what would otherwise be an ammonium flux coming from the sediments into the water column. We do see evidence of nitrification also, even over the short time period, we see nitrification of this ammonium. That's likely why you get this plateau in ammonium at lower cell concentrations. And then last but not least, we wanted to look at if we had microcystin toxins fluxing as well. We did see microcystin toxins fluxing in a couple cases, but as you could see over here, now microcystis in the ambient water is our independent variable, and now our toxin flux is our dependent variable. And what you see is that the toxin flux is somewhat proportional to the amount of microcystis present in the bottom water. And so, it's kind of inconclusive as to whether we have an actual microcystin toxin flux or whether we just have trapped microcystis inside the chamber that's generating new toxins. You can also see evidence, here's that scoop with ammonium. We have oxygen being consumed by the sediment. You can see here some of the highest time points. This is the Redfield ratio, by the way. But here is your highest time points. You see when we have microcystis present in these green data points, you don't have the ammonium outward flux that you would expect based on, at least the oxygen being consumed. So, definitive evidence that you have microcystis taking up the ammonium in the bottom water. Now, this is new work that I want to talk about. We just got the system, and we're going to start it tomorrow. Now, this is something that has been done in Lake Erie, actually, by our collaborator, Tim Moore, Kai Zervagal, who's faculty at UNH. We're deliberately taking cores and inducing resuspension on them using a propeller. And we want to see, we want to be able to link the water velocity, the bottom water velocity that's induced on a sediment, to see how much of the sediment is actually resuspended and how much of the pore water is resuspended. And we think this is going to be critical information for the modeling. And then, hopefully down the road, we could even just monitor turbidity in the water column and then perhaps back calculate the depth of the sediment that has been eroded. And if we know the nutrient content in that sediment, let's say from the model or from measurement, now we can infer the resuspension-based nutrient delivery to the water column. We're also trying a new system if FedEx, if it clears customs in time for Friday. Don't get me started. This is it's called a sedimeter. It's basically a bunch of turbidity sensors, and you install it in this sediments, and it monitors the water depth over time. So I just found this on social media recently, and it's so cool because it's something that I've been wanting to design for a long time, and it's actually already been made. So we're going to try to monitor resuspension in situ using this. We're also going out every, I guess we'll be doing this quarterly, these really comprehensive 16-hour on-water days. So now this is that same diagram as before, but I did invert it horizontally just because it better shows what I want to sort of indicate. What we're going to try to do is go out right after a storm event and install a bunch of in situ instrumentation to monitor the bottom water, monitor that resuspension using that sedimeter, other stuff like that, install the AUTOHOLO. And we want to do this right after a storm. And then let's assume it takes a few days for things to settle out, for this Microcystis to start doing its trick, where it may be migrating over day and night periods. And we're going to go out for a 16-hour day right after the storm, and then we're going to wait until a nice quiet period, let's say 4 days later or so, and go out again for a 16-hour period. And as you can imagine, it's crazy logistics to try to plan, but we think it's necessary to really answer the main driving questions that we need to ultimately parameterize the model. So in terms of the sediment work that we're going to be doing, there's phosphorus and there's nitrogen. I already showed you sort of some of that work from the electrochemical profiles. We're just going to be doing some more of that to try to really identify, the iron controls on phosphorus cycling. But then we're going to look at a new process, and I think this is the first time I'm introducing this. But there's a new biogeochemical process called FIAMOX, iron-coupled ammonium oxidation. It's already been demonstrated to be very important in Lake Tai or Lake Taihu in China, which is, it's very similar to Lake Okeechobee, but we even have more iron in our system, so it should even be a stronger process in our system. Because we don't have oxygen in our sediments, and because we don't have much nitrate or nitrite, that should be, in our sediments, we shouldn't have, in theory, there's really no other pathways that could result in that ammonium removal over time that we think that we see. The whole idea is that ammonium is directly oxidized to nitrate using iron oxides instead of oxygen, and then it can ultimately be denitrified after that. And so we'll be collaborating with University of South Carolina, Annie Bourbonnet's lab, to try to use or to conduct some incubations using nitrogen isotopes to try to see if this process is the main mechanism by which we're removing ammonium from the pore water. But sort of, the moral of the story, what I really want to say is that the long-term eutrophication of the system, it may have effects on the redox environment, right? So if you have a lot of organic input, a lot of nutrient input, you're going to create a more chemically reducing environment. You're not going to have iron oxides. They're not going to be favorable to remain in their oxidized form. They're going to want to be chemically reduced. When they decrease in concentration, you can also increase their saturation, or the saturation with respect to adsorbed phosphate. So now, you really have, well, let's just say that iron oxide can essentially be coupled or can be tied to both these long-term increases in pore water phosphate and pore water ammonium if you've now affected your overall iron oxide concentration. So this is really sort of the ultimate synthesis of what I want to try to show, or at least what I think is happening, from the geochemical perspective. In the case that iron oxides are really one of the primary protections on the system. So this is my last slide. I'm just synthesizing, what we found in Okeechobee versus at least what I think, based on my very poor understanding of the Lake Erie literature, at least the Western Basin. It's much deeper. I shouldn't say much deeper. It's not that much deeper, but it's deep enough. It's over twice as deep. It's enough to probably affect the ability of resuspension. I showed you how easy it is for sediments to be resuspended in Lake Okeechobee. It's probably not that easy in Lake Erie. So, right off the bat, that's probably a big difference. Sediment oxygen, it only penetrates 1 millimeter in Lake Okeechobee, penetrates up to 50 millimeters in Lake Erie, according to Matisoff et al. And I really enjoyed this paper. I thought that was an awesome paper. I didn't realize y'all were doing such good work. Well, I shouldn't say such good work. I should say such advanced sediment techniques up there in Lake Erie. So that was a pleasure to read. We know that iron cycling is critical in Lake Okeechobee. We see correlations between phosphate in the pore waters and dissolved iron in the pore waters, but this is not the case in Lake Erie, I believe, according also to this Matisoff paper. We have a very fast turnover time with respect to our sediment diffusive flux, at least those measured based on the sediment core profiles. We're talking, 1 to 7 days. So our sediment fluxes are very important. Our internal loading is very important relative to external loading in terms of the nutrient budgets for the lake. We don't detect sediment phosphate fluxes under aerobic bottom water conditions. I think you do detect some in Lake Erie, but you detect much more under hypoxic conditions. Casey Godwin, my primary contact up there, he had a really cool study where they used in situ bottom water phosphate sensors, monitor over time, and you can see very clearly that when you have the onset of hypoxia, you get a flux of phosphate from the sediments. It's been shown in Lake Okeechobee cores that you can induce a phosphate flux. If you induce hypoxia, the only thing is that we don't really ever get hypoxia events in Lake Okeechobee because it's so shallow and so easily mixed. We think that nitrogen co-regulates the Okeechobee algae. We saw the bloom really begin in the wet or prior to the wet season, the bloom begins. So it seems like this internal loading is perhaps more important in Lake Okeechobee than it is in Lake Erie. And we also see this timing mismatch between sediment respiration and ammonia maximum that suggests that the nitrogen's being actively removed over time. And the last thing that I didn't mention yet is that we did look at the sediment cysts, Microcystis cysts in the sediments, and we rarely actually see them in the lake. And when we do see them, they don't seem viable. So, we may not really have this year-to-year seed stock, which to my understanding, I think is something that does happen in Lake Okeechobee. And, that's maybe the cause as to why you at least have the seeds present to start the blooms from year to year. So to kind of wrap it all up, I wanted to show that, this popped up on my newsfeed that even this past year, the seasonal model undercalled the Lake Erie, it underestimated the severity of the Lake Erie algae bloom. So it seems like you guys in the Lake Erie area working on this system, you still have a lot to learn about the geochemical processes. In our situation, we're even further behind, right? We only installed, not just us, but anybody in Florida, really only installed in situ continuous monitoring systems beginning in 2020. There's no spatially resolved numerical biogeochemical model except the one that, we're in early production on ourselves. There's no diagenetic model like what I saw in Matisoff et al. So we're just beginning. We're a little bit behind, and hopefully we can learn a lot from you guys and likewise. So I'm going to stop there. Happy to take questions.
Thank you so much, Jordan. That was a wonderful and very interesting presentation. I would like to invite our attendees to enter questions that you may have for Jordan into the question box. And we will be wrapping up this webinar in about 5 minutes, but I will be putting a link to a Google Meet that people are welcome to switch over to at 2 o'clock for an extended conversation with Jordan. So I will move to our first question, which is from Casey Godwin. Casey says, thank you for that interesting presentation. Can you please describe the stakeholders connected to Lake Okeechobee and any interactions you might have with them through this research?
Well, so this was funded right by the mitigation program, a new mitigation-based program. And so, in terms of commercial stakeholders, at least directly tied to the algae blooms, we have pretty intense dialogue and relationships with these folks. everyone is trying to cash in right now. Everyone has a solution to solving the algae bloom problem, right? So everyone wants to try out these techniques, and they contact us a lot. We have a lot of folks who want to sort of tie in their mitigation tests or efforts along with the work that we're doing. In terms of the stakeholders, well, we have our relationship obviously with the department, with DEP. One example that I meant to mention was that the Pahokee Marina, when we saw that ammonium down in the pore waters, the whole idea was, do we want to dredge this? That's what they were asking us. Would it make sense to dredge this marina because it's a nutrient hotspot? And what we concluded was possibly, but even in our control site, our reference site that was outside the marina a little bit, we also saw very high ammonium concentrations. So it seems like there's sort of some leakage, I guess, from this marina hotspot. In terms of local stakeholders, just from, let's say, the community, I guess, to be honest, we haven't interacted all that much beyond just the occasional presentation and whatnot. We haven't really formed, I guess, any direct interest to try, people trying to use our work to try and perform some public good. I guess we do discuss with the fishermen a lot the results of the algae bloom aerosol work. We plan on publishing that, and we're constantly in contact with the people that are out in the lake all the time with respect to if they're breathing in algae toxins. But beyond that, nothing else really rings a bell immediately, but I'll try to think, in this way we can discuss it afterwards. Thanks for the question.
Yes, thanks for the question, Casey. If anybody else has a question, we have about 2 minutes left before we will jump over to the Google Meet. And I did put that link in the chat box. I also put the link to the page on the Sigler website where the recording for the webinar will be posted in by the end of the week, in case you want to share that with colleagues or view it again yourself. Okay, so question from Tom Bridgman. He says, thanks for the excellent talk. You mentioned spat bags. How well did those work?
Thanks, Tom. Yeah, so the spat bags was something that we added in the latter half of the year. So let's say, I think, beginning January of last year, and they worked well. It seemed to mirror the in-water toxins. We lost quite a few of them, I know, but yeah, I mean, they seem to mirror the in-water toxins, and we actually have samples pending to send off for LCMS analysis, because I'm just basing what I just said on just ELISA analysis, just for microcystin LR. So they work pretty well.
Great. Thank you. We've got time for one more question if anyone has one, and if not, we will switch over to the extended discussion on Google Meet. Okay, well, we're right at 2 o'clock and I don't see any other questions or hands raised. So Jordon, thank you very much for the great presentation. It was really cool to hear about all of the connections between Lake Okeechobee and Lake Erie and some of the similar work we're doing. So thank you very much for that. And
thank you, Mary.
Yep, and thank you everyone for attending. And again, I encourage you to continue the discussion on the Google Meet. Thanks, everyone.
Bye-bye.